So I'm Megan, and this is my partner in crime, Kiera. And we've been working with gold nanoparticles, and we've brought a little bit to show you about it. But gold nanoparticles help in finding a detection of cancer when you go through an MRI scan. So she's going to tell you about the importance of the kudzu plant.
Okay, so basically kudzu is a vine and it has a root, a flower, a leaf, and a stem that can be used in many Chinese medicines. Kudzu was introduced to the United States from Japan but is native to China. Kudzu is very popular in Kentucky and many southeastern states, which is why we chose this plant. But not many people have experimented with kudzu in the process of gold nanoparticles, but we figured it would be a good substitute for the sodium citrate, which is a toxic solution, and you have to intake this whenever it has to be detected through the MRIs, so toxicity levels are not good.
So the gold nanoparticles, this is an example of the gold nanoparticles. We're trying to create a solution that's less toxic than the one that's intake or consumed now. So how it works is the gold nanoparticles, when consumed, the gold actually attracts to the cancer cells, and that's how it shows up in the MRI. And the way that we've put the kudzu into our solution is we have taken the extract from both the stem and the leaf and mixed it with a solution of 180 milliliters of diluted water and 20 milliliters of hydrogen tetrachloride. And in this, we boil it on a hot plate, and after about 10 minutes or so, you see a change in color. And the change in color of it goes from a brown color, like a brown yellow, and after 10 minutes turns into like a purple or a ruby red. And that shows that there are gold nanoparticles present in the solution. So next we'll talk about the data that we've recorded.
So far, we've not been able to record much data because our spectrophotometer broke. But basically, you can tell that it's gold nanoparticles whenever it has this amp right there. And it is a 0.3 is the maximum we've had, and that's with 6 grams of kudzu leaf solution and 200 milliliters of distilled water. The wavelength of this is at 520 nanometers, and that's pretty much all the data we've gained so far from this experiment.
We're waiting on our photometer to come in. And these are the things that go into it so that the light can be shown through it to get our data. And so plants are used to get gold nanoparticles in replacing the toxic solution we have here. And basically any herb that can be used as like a natural medicine can be used in this project, or so we've seen. So we decided to use kudzu, and it's shown really, really, really good data. And we have yet to get our final solution that we need, and she's going to tell you a little bit about how we're going to get there.
So basically, in order to get the toxicity levels down, we are going to use a process called electrolysis. And that's whenever it breaks down it breaks down the gold, which is the gold rod will be our anode, and then the copper rod will be our cathode. And it'll break down those particles, and it'll mix with chlorine and our solution, and it'll create gold chloridal aurate, or gold mixed with the gold atoms. And it's a substitute for the sodium citrate, which is very toxic and cannot be intook within the body because it's toxic. And so it is, in some things, like it's proven that electrolysis will help the toxicity levels go down.
So after we're done with the electrolysis, we hope to see that not all the toxicity, but most of the toxicity of our solution is down. And this will be able to be consumed by a cancer patient and like by the mouth even. And if they have an MRI, we'll be able to see exactly where their cancer is in the cells. And we're going to study this at Louisville's Speed Lab to use the scanning tunneling electron microscope. And that's what we have.